everyone, and welcome to today's webinar, From Development to Regeneration, the Role of Epicardium. I'm Christina Jewell of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific, Gibco. To learn more about our sponsor, visit thermofisher.com/Gibco. Now, we encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We will answer as many of your questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have any trouble seeing or hearing the presentation. It is now my pleasure to welcome our speaker, Jun Jie. For a complete biography on today's speaker, please click on the presenter's window at the top of your screen. JJ, welcome. You may now begin your presentation.
Thank you, Christy, for the kind introductions. Hi, everyone. It's my great pleasure to be here to share some of my work on these topics. And let me introduce myself briefly. I actually earned my bachelor degree in biomedical science from Universiti Putra Malaysia. I also did my doctoral degrees in the University of Oxford. At the Department of Physiology, Anatomy and Genetics. I was also a visiting research fellow at King's College London, and I did my postdoctoral research fellowships at Mass General Hospital, Harvard Medical School. I have served at Universiti Sains Malaysia as a senior lecturer since 2011, and I have now got promoted to the associate professor until today. I would like to actually share this slide as the first slide about the disease. Heart disease is still the main cause of death globally, and is accounting for estimated 17.9 million deaths annually according to the World Health Organizations. And ischemic heart disease is actually the main cause of heart failures. And in Malaysia, 70% of the total medically certified death is actually caused by ischemic heart disease. And why the disease is so tricky, the ischemic heart disease is also known as the coronary artery disease, which the blockade of the coronary artery caused by the arteriosclerotic plaques, as it stops the blood supply and also causes ischemia in the myocardium. This makes the heart respond to the damage by undergoing some of the changes in the structures and also the functions. The process modeling. Still can see from these MRI scanning videos, this is a very healthy human heart. The ventricle is contracting very forcefully, as you can see over here. On the right is the failing heart, where you can see a majority of our myocardial wall is akinetics, and the contractions of the ventricle chamber is not as forceful as compared to the normal heart. And this remodeling actually caused the heart to become enlarged and eventually fails to keep up with the outputs. The disease is very detrimental because upon the initial diagnosis of heart failure, 75.5% of the patients are able to survive the first years, and the rate deteriorates down to 45.5% only in 5 years, and only 25.5% of the patients survive after 10 years. So in a lot of our efforts in the past decades, we are looking into the cell type that we are able to repopulate the cardiomyocytes or the heart cells that die in the process and cause the remodeling. And what we are aiming for is to have some of the cells to repopulate the regions and regenerate the region, and so we can restore the contractile functions. To look into the different cell types ranging from mesenchymal stem cell, bone marrow-derived stem cell, epithelial stem cells, to the more closely related cardiac-derived stem cells, and also the embryonic stem cells. And in my initial career, I was looking at the endogenous cardiac stem cells, where we were hoping to stimulate the stem cell that actually resides in the heart to do the regenerations by activating them and hoping for these endogenous cardiac stem cells to differentiate into cardiomyocyte, and so we have more of these formations of the new myocardium, and together with the blood vessel that are able to actually help and contribute to the overall contractility of the hearts. Unfortunately, the initial promising study that we found in the preclinical settings in the laboratory has later on been proven to be less promising in some way. As you can see, a lot of our study in the early stage, early research were supporting the evidence that the cardiac-derived or bone marrow-derived stem cells are able to contribute to the formation of the new heart cells, but not with more sophisticated or advanced laboratory techniques that are able to do the genetic tracings of the cells. Those cells were found not able to form the functional cardiomyocyte in the adult hearts. So in the community, a consensus almost reaches that most of the adult stem cells that we send, although we see some of the improvement after the injections of these cells into the infarcted myocardium, these improvements were mainly attributed to the paracurrent effects rather than the formations of the new myocytes. And so we still need some sort of cells that are able to form functional cardiomyocyte in order to successfully regenerate the functions. So we are looking into the pluripotent stem cell. Pluripotent stem cell is the only promising cells that are able to make functional cardiomyocytes. And the earliest pluripotent stem cell that we know is actually from the embryonic stem cell, which is harvested from the embryo, the fertilized embryo of the blastocyst. So the inner cells muscle of blastocysts are able to differentiate into all types of cells that you can name in the human bodies, but it also get complicated by the ethical issue behind the use, whether or not fertilize the human eggs and destroying by harvesting the cell is equivalent to killing a human entity. So in 2008, Shinya Yamanaka, the Nobel laureate of Physiology and Medicine of 2012, introduced the technologies called reprogramming, and the sort of reprogramming allow the somatic cell to be reverted to the pluripotent stage, and these cells are able to behave exactly like the embryonic stem cells and are able to differentiate to all types of cell type that we already know about from the embryonic stem cells, including the cardiomyocytes. And in our laboratory, we also try reprogramming the frozen cord blood, which has been cryopreserved for more than 10 years. We harvest the CD34 and reprogram it into iPS cells. And we are using the Thermo Fisher's Epi5 reprogramming kits, and we successfully turn the colonies, the cells into the pluripotent cells, induced pluripotent stem cells. These cells are able to express all the pluripotent genes, and including the differentiation of the cardiomyocytes, as we show over here. We use a very simple and direct but known cardiac differentiation method that actually employs the temporal stimulation of the Wnt pathway, modulations of the Wnt pathway that directs the cardiomyocyte differentiate. We started off with the Wnt signaling activations at the early stage, and then continue by the Wnt inactivations at the later stage. We deprive the cultures at the early stage, at least 7 days. Culture deprived from the insulin stimulations. We believe the insulin stimulations will actually impede the cardiac differentiate at the early stage. So in our culture, we are able to turn all these iPS cells into cardiomyocyte at a super high efficiency, and it can show, as you can see from the well that we actually capture over here, and most of which were also expressing the ventricular specific marker MLC2V. So differentiating cardiomyocyte is no longer one the major challenge. The challenge is whether we are able to actually make the cells useful for therapy or for any of the in vitro testing that can actually resemble the human myocardium. And at the moment, most of the cells, most of the cardiac tissues that we are able to engineer in the laboratory are consisting of very simple cellular compositions or a very simple formulation of the matrices. It has a significant lack of the cellular and structural complexity, some of which cardiac construct that we engineer in the laboratory is weak to be handled by surgeon for implantation, so to speak. So in order to look into improving the cellular complexity and also the structural complexity, we want to know how the whole process is formed during early heart developments. And what we are interested in is these particular organs called pro-epicardial organ, as shown in these green structures over here that actually sits at the veins for the looping hearts. And these pro-epicardial organ actually derive the epicardial cells that actually adhere at the outer surface of the heart and form the structures called epicardium. And why is it so important is that if we, some of the study actually shows that knockout of this WT1 is this in these epicardial cells will actually cause a lot of changes in the heart structures. As you can see is in the wild-type hearts, the heart is, the myocardial wall is very compact with a very nice layer of epicardial surface, which is expressed in green. Knockout of the WT1, you can see much less of the epicardial cells. On the outer surface, there's no invasions or integrations of the epicardial derived cells into the myocardium, and you can see the cells is actually much less in both at the ventricle and the myocardial wall. Here we can, the data actually shows that the proliferating cardiomyocyte is much lesser in the knockout. And also when we look at the coronary artery coverage in the embryos, the vessels is also much lesser in the WT1 knockout. So that actually means that epicardium formation is very important for myocardial proliferation and compactions, and also the blood vessel formation in the early stage. And we sort of like understand the epicardial cells is important in these instances and how we can even use it. Maybe if we are able to use it in a tissue engineering, we are able to actually engineer a myocardial tissue that is closely mimic the human myocardium. The easiest source of epicardium would be coming from the adult tissues. If you are able to harvest the adult epicardium, you are able to isolate the epicardial cells, we should be able to use it directly. Unfortunately, the epicardial cells from the adult source as compared to the fetal one has fundamentally different function and capability as shown in these papers. So we have not actually seen any of the publication that just shows that adult epicardial cells are able to stimulate cardiomyocyte proliferations and also blood vessels formation in vivo or in vitro, so to speak. So we believe that we need to actually look for the epicardial source from the fetal tissue. But harvesting epicardial cells from the fetal tissue also complicates the real-world practicality, because we have very limited sources of tissues that we are able to harvest those cells and expand them in the laboratory. So the beauty of using induced pluripotent stem cell or the iPS cells is that we are able to actually generate or make these cells from the embryonic stems, from the pluripotent stem cells, and expand them for our use. So we look into the signaling pathway that is involved in the epicardial development, the early epicardial development, and we actually direct the differentiation of the induced pluripotent stem cell by initiating the lateral plate mesoderm formations using prolonged Wnt activations in the early stage. So we are able to direct the IPS differentiation to lateral plate mesoderm, and from lateral plate mesoderm, we include the stimulations of three major factors, which is the BMP4, retinoic acid, and VEGF. BMP4 and retinoic acid are the keys morphogenes of directing these pre-epicardial cells, and the VGS is mainly for inducing the proliferation of those cells as in our hand. So as you can see, during the entire differentiation process, upon all these stimulations, the pluripotencies of the cell had decreased, and the cardiac lineage markers has not changed except the troponin T gene expressions. Although we see the significant expressions of the troponin T in these cells, we don't see the expression of the proteins in these cells at these levels. However, we actually see all the significant changes in the epicardial-related markers such as WT1, DBX18, SEMA3D, SCX, TVX5, and BNC. So all these markers indicate the identity of the pre-epicardial cells that we obtain. We also stimulate the differentiated PECs or the pre-epicardial cells to test their capacities and the capability to undergo EMT. This is one of the very important characteristics of epicardial cells. And upon the stimulations of the factors, the related factors such as TGF-beta, BFGF, or the combination of both, we are able to induce epithelial mesenchymal transitions. That's upregulations of the smooth muscle cells, smooth muscle markers, such as smooth muscle actins, DDR2, TGF, these are prostaglandins expressions, and also TGF21 and vimentin actually indicate the fibroblast markers. We also see a significant upregulation of the CD90 expression upon all these stimulations, and the most upregulations of this mesenchymal gather happens upon the stimulations of BFGF. We also acknowledge that the pre-epicardial cells that we generate are quite dynamic. It requires constant inhibition of the TGF-beta using the SB small molecules. The SB431542s is what we can actually keep the pre-epicardial cell at the epithelial stage. The fundamental characteristic of the epicardial cells is that if we co-culture with the cardiomyocyte, they has to be have the capability to mature and actually work together with the cardiomyocyte. And we have seen the upregulation of the E-coherence and also some of these epithelial markers as a uroplakins, uroplakins 1B, and also the alpha-4 integrin, which is also expressed in epicardiums. We also see some of the upregulations related to the smooth muscles and also the gap junctions that we already know in the epicardiums. What is more important characteristics that we want to actually have for our cells that we actually generate in the laboratory is the functions of these retinol aldehyde dehydrogenase. These enzymes is actually responsible for the retinal acid secretions in the paraepicardial cells. So to take this characteristic, the characterization further, we actually compare with the known epicardial cell, which has been derived by other laboratory and has been published previously. So we obtained the cells line from Sons Palas's lab, which they derive the epicardial cells from the H9 embryonic stem cells, and we compared the two lines with our lines. What we actually see is that our cells, pro-epicardial cell at day 7, freshly differentiated one, fundamentally or genetically quite different from the epicardial cells that they actually provided. But upon one passages of these pre-epicardial cells, we are able to see some of the majority of the changes of the genes is quite closely similar to the epicardial cell that we already know. What we also observe is that the PASHAS1 epicardial cell that we have in the laboratory has more of the genes are regularly related to heart development, circulatory system development, or mesenchymal development, etc. All these other characteristics or the functions that we want to see from the pro-epicardial cells compared to the more differentiated one. So when we actually put the cells in a more defined compartments inside the cell culture insert, where we actually put the cardiomyocyte in the middle and also have the pro-epicardial cells on the outer layers. The pro-epicardial cell, we will tag it. We tag the cells with the mTuris, and we allow the cells to actually migrate and invade into the cardiomyocytes regions where we actually seeded earlier that express venous. So we can actually see there's a lot of spaces in between the cardiomyocytes, but over time when the pre-epicardial cell invade the regions and the cells get more compact and less region in this cardiomyocytes seeded area. And more importantly, these cardiomyocytes are still contracting synchronously throughout the whole cultures than before. And you can see there's a lot of epicardial cells. The epicardial cells actually get differentiated or undergo MMT expressing WT1. Most of it will still retain the WT1 expressions in the co-cultures. So we take this observation a step further to look into how the pre-epicardial cell affects the cardiomyocyte in the co-culture settings. We co-cultures the two iPS-derived pre-epicardial cell, which is tagged with mTuris, and also the cardiomyocyte, which is tagged with venous, so we can clearly distinguish the two populations. And upon the co-cultures of the two cells and maintaining for up to 8 days, we see the formations of cardiomyocyte island, individual island that's not connected with each other in the early stage, but they still remain spontaneously contracting in the cultures. However, if you actually prolong the cultures up to another 16 days for another 8 days, you can see these cardiomyocytes started to connect, and then they form a very nice network without all these islands anymore. So in some way, the cardiomyocyte migrates and they start to actually form network in the core culture. We don't see it in the monolayer cultures as shown over here. These observations also come with the reduced substantial reductions in the cardiomyocyte area coverage, and also the significant increase in the aggregate height. So we sort of believe is that the pre-epicardial cell in the co-culture setting are able to compact the cardiomyocyte to a certain extent. And this network of cardiomyocyte in the pre-epicardial co-cultures are able to show synchronized excitation contractions coupling under unstimulated and also point stimulated conditions with 20 volts with 0.6 milliseconds to 0.2 hertz powers. So we also sort of like look into what are the functions of this pre-epicardial cell could induce. One of the very important features that we want to actually see from pre-epicardial cells is that whether or not these cells are able to stimulate cardiomyocyte proliferations similar to what we have observed inside the embryos. So we also study the expressions of this EDU in cardiomyocyte after 6 days of co-cultures, and we see a very significant increase in the proliferating EDU positive CT&T positive cells in the co-cultures. And when we actually looked at this data much closer, we also realized that there's IGF-2 involvement in the process. So we use lucitinib, which is the inhibitor for IGF receptors. There's a dose-dependent decrease in the cardiomyocyte proliferation, and stimulation with the IGF-2 alone also sees a dose-dependent increase in cardiomyocyte proliferation. And what we also do is that we also look into the IGF-2 expression inside the conditioned medium after co-culturing the two cells, and we see a significant increase of this IGF-2 inside the conditioned medium after day 6. And this is more significant when the cardiomyocyte is present. And when we increase the cardiomyocyte number, we also see the increase in the IGF-2 expression inside the pre-epicardial cells. And what we also did is that we hypothesized whether or not the direct contact of cardiomyocyte is required for the expression of IGF-2 in these pre-epicardial cells. We actually do the insert cultures. In the control, we put the cardiomyocyte inside the inserts, and we have a pre-epicardial cell at the bottom of the walls. And what we actually assess is that we actually take the pre-epicardial cells for the IGF-2 RNA analysis. And what we see is that in the co-cultures setting when the cardiomyocyte was present, there's a expression of the IGF-2, but when the cardiomyocyte was absent, there's a reduction or very significant lower IGF-2 expression were present in the pre-epicardial cells. And what we also see is that when we inhibit the RNA, when we inhibit the retinoic acid receptors, the RXR alpha receptors on the cardiomyocyte using BMS, small molecules, we also see a significant reductions of the IGF expression inside the pre-epicardial cell. So we believe that the PECs, the pre-epicardial cell, express the IGF-2 in response to the stimuli of cardiomyocytes, and it's via the retinoic acid signaling. We also look into some of the functions of the cardiomyocyte in the pre-epicardial cell co-cultures. I will call it PEX cells co-cultures from now on. We see, as we know, it has the ability to aggregate cardiomyocyte, as shown in the aggregate height measurements over here in the first graph. That, as compared with the HUVEX and the human cardiac fibroblasts, the cells basically overtaking the whole entire cultures and in some way kills the cardiomyocyte. These cardiomyocytes were not able to contract in the co-cultures. We also see the much upregulation of the much improve in the atrial strength in the PEX co-cultures, PEX and cardiomyocyte co-cultures. And we also see the modulus, the young modulus, the stiffness of the gels is also increased in the PEC co-cultures, but not to the extent of HUVEX co-cultures. Co-cultures actually cause more of the stiffness inside the co-cultures. We also see the improvement in contractilities and also the calcium handling measures by the amplitude, V-stroke, and decay. We don't see much of the increase in the amplitude of the calcium signals, which is not driven, but we see the upward trends. And this is also has a significant difference as compared to the HUVEX and also human cardiac fibroblast co-cultures. We see very significant V-strokes, V-upstrokes, but not so much of a V-decay. But again, we also see a much significant difference as compared to the HUVEX and also human cardiac fibroblast co-culture. This also indicates that PECs are able to improve calcium handling in cardiac co-cultures. We also take a step further to look into how the PEC would affect cellular maturity of the cardiomyocyte. We look into the mitochondrial content in the cardiomyocyte. We see a significant increase in the mitochondrial content in the cardiomyocyte. We see a significant changes in the sarcomere length in the cardiomyocytes, but we don't see much of the improvement in the cell size. And again, this may be due to the duration of the experiment we did in the cardiomyocyte PEC co-cultures. If we are able to make we prolong the experimental periods, I think we should be able to see the difference as well. We also combine the two differentiations since the lateral plate mesoderm cells are able to derive cardiomyocyte as well, which we have already proven in our experiments. We use the lateral mesoderms and we combine the two differentiation stimuli that we use for cardiomyocyte and PECs. When we are able to have induced co-differentiation to have both cell type exist in the single cultures. What is more intriguing is that they don't randomly form and grow separately. They actually show some of the defined spatial cellular arrangement, like what we show over here. The cardiomyocytes tube that actually form in the middle, covered with a very nice layer of the pre-epicardial cell expressing WT1. And we also sort of like imagine if these are real pre-epicardial cells so we also imagine that if these are pre-epicardial cells, they should have retained the ability to adhere and form an epicardial layer on any of the cardiomyocyte surface. So what we did was we produce the 3D cardiomyocyte spheroid. At the same time, we produce the PEC spheroid. We put it into the spin culture and co-culture them. And we don't see any of the smooth muscle layer presence in any of the cardiomyocyte spheroid culture, single cardiomyocyte spheroid cultures, but we do see a single layer of smooth muscle positive cells layer on the surface of the cardiomyocyte spheroid as shown over here. Besides, we also notice that there's a formation, structural formations within the spheroid that we co-culture the two spheroids, where they actually form a lumen-like structures. These lumen-like structure has a very similar cellular arrangement like the vessels. So it has an inner layer of the V-coherent positive cell with enveloped by circulators or covered by outer layer surface of the smooth muscles. So we believe that in some way the pre-epicardial cell actually enhance the morphogenic complexity in the 3D cardiac aggregates or spheroid cultures. So there are a lot of protocol has been published comparing different stage of epicardial cell that they can derive from human pluripotent stem cells, some of which we're using embryonic stem cell, and as you can see, most of which actually have a much longer or complicated protocols or steps to derive the epicardial cells. And we are the only one that actually have a very simplified differentiation versions to derive the pre-epicardial cells using the very simple direct stimulation of factors and able to actually produce them in 7 days. And we are also the first one to actually demonstrate that these cells that we produce are able to secrete IGF-2, they are able to induce cardiomyocyte proliferation, they can actually aggregate cardiomyocytes and work together with the cardiomyocyte in the single co-cultures. So in summary, the pre-epicardial organ-derived cell, or the transition intermediate cells prior to forming the epicardial cell actually has a very significant role in myocardial regeneration, and which are likely the critical factor that will facilitate the creation of thick myocardial tissue. This is one of the challenges that the community would like to overcome, where we can have more vascularized tissues that are able to give significant contraction forces upon implantations to any of the myocardial infarction patients. We are the first to demonstrate the two fundamental functions that human pre-epicardial cells are capable of, which is the ability to form the epicardial epithelium on the outer surface of the heart. We are also able to actually show that the cells are able to express and secrete insulin-like growth factors 2 in the co-culture, and these aspects recapitulates the in vivo development. We are also able to demonstrate the PECs are functional. They are able to interact with cardiomyocyte and enhance the function and the structural organization in 3-dimensional cultures. In the PEC cardiomyocyte microtissue co-cultures. So in my future work, I would like to explore the use of these PECs in cardiomyocyte therapy and see how these cells can be harnessed for therapy in treating myocardial infarctions. We also want to explore looking at how PEC involved in vessel engineering and generates more vascularized thick cardiomyocyte or cardiac tissues. We are also looking into how the role the PECs induce the cardiomyocyte maturations and whether or not there are some other cell types that they are also playing a role in this process. And the whole idea of making these cells is also stem from the original idea of how we can actually improve the process of recellularizations. On the left, this is the white whole organ. It's actually the completely decellularized porcine hearts. We are sort of trying to improve the technologies of how we can engineer or put the right cell into the right space back to the scaffold. And so ultimately, we are able to engineer a human functional hearts on the right. So with this, I would like to thank my mentor, Dr. Harold, has been very supportive of this work. Dr. David Milan, Jackie has been working side by side with me on this project together with all the colleagues from our labs in Mass General Hospital and Harvard Medical School. Dr. Catherine Hassan has been helping with the construct and assessment of the heart's functions in the PEC co-cultures. Dr. Kinhua Lin has been helping me with the RNA sequencing analysis. My student in Universiti Sains Malaysia working on generating the iPS cells from the couplets, and this project was supported by Harvard Stem Cell Institute, Cell Fabricant MGH Research Fellowship, as well as the Ministry of Higher Education Malaysia. With this, thank you for listening, and welcome if there's any questions.
Thank you, JJ, for your informative presentation. It is now time to start a live Q&A portion of our webinar. Now to our audience, if you have any questions you would like to ask, please do so now. Just type them in the Ask a Question box and click Submit. Okay, JJ, so we have a few questions already in, so I'd like to start with this one. What are the potential applications of PECs in tissue engineering and therapeutics?
I think for PECs, I would like to actually use it and see whether we can actually harness the potentials like what we have seen in the embryonic developments, the heart developments, in inducing the cardiomyocyte proliferation in situ, and also whether or not the combinations of these cells together with the cardiomyocyte therapy would actually help with the cardiomyocyte engraftment and also stimulation of the angiogenesis or coronary vessel formations in situ as well. So we are very excited to sort of explore into whether or not sending this cell into the infarcted heart will actually helps the cardiomyocyte therapy in some way, and also ultimately the overall heart functions and the outcome.
Thank you, JJ. And I think we can squeeze this one in as well. What kind of future work are you planning to do for a more comprehensive understanding of not only your research, but also its implications?
Sorry, was try to think. Right. Thanks for the question. I think that's a very good question. We are currently looking at how we can include the pre-epicardial cells into cardiomyocyte therapy, especially on the numbers of the cell that we want to actually include in a single dose of cardiomyocyte. We want to send it into the infarct regions. We want to see how the cells become when the cells were inside the infarcted heart, so whether or not they become more proliferated, or they are doing more of the jobs to consolidate the heart architectures and prevent remodeling in some way with the presence of cardiomyocytes. We're also exploring using these pre-epicardial cells into engineering of the blood vessels and see whether we or not we are able to increase the thickness of the tissue that we are making, and how these cells are able to attract, for example, endothelial cells in the structure and form a more spontaneous from the network of the vessel inside the construct. So these are the main interesting aspect that research that we are looking at right now.
Thank you, JJ. Now, do you have any final comments for our audience?
I would like to say hi to my mentors. Harold, hi. I finally presented this work together with Thermo Fisher Scientific. Thanks for the platform. Hi to my team in Universiti Sains Malaysia. Thanks for all the opportunities it has provided to me to enable this work to be done. Right.
And thank you again, JJ, for your time today and your important research. We'd also like to thank LabRoots and our sponsor, Thermo Fisher Scientific Gibco, for underwriting today's educational webcast. Now, before we go, I do want to thank our audience for joining us today and for their interesting questions. Questions we did not get to today, and also those that are submitted during the on-demand period, they will be addressed by our speaker via the contact information you provided at the time of registration. Today's webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. That's all for now. Thank you so much for joining us. And JJ, thank you again. Have a great day.
Thank you, Christine, for hosting me. Thank you. Bye-bye.
Bye-bye, everyone.